actually have sodium which can leak into the cell. Okay, if the sodium can leak into the cell and this pump is not working, how is that possible? There are many pathological situations. For example, when hypoxia happens, what happens in that? The pump stops working or reduces the working, or lesser number of pumps are working. And what would happen is sodium would start diffusing into the cell. When the sodium would start diffusing into the cell, it would pull water with it. So we'll talk about that in a bit, that how do the osmosis work. The basic idea here was to actually see the compositions. So if the water would actually follow sodium inside the cell, what would happen to the cell? Cell would swell. Correct? So sodium going into the cell, it is taking the body water with it inside the cell, and the cell is going to start swelling, and that is what happens. You know that in the hypoxic situations, it is possible that the cells swell up. They swell up because their sodium potassium ATPase pumps are not working, or working less than normal. So sodium potassium pump is really important. What it does is it throws the sodium out and it brings the potassium in. So as a result of that, of course, the sodium concentration outside the cell is more and potassium concentration inside the cell is more. Right? Sodium outside the cell is more, inside the cell it is less. Potassium inside the cell it is more, outside the cell it is less. Why? That is because of this pump. So don't forget this. So sodium outside 142. Look, I'm saying 142. I have 142 here, I have 140 here, but let's say for the time being, extracellular fluid 142 milliosmoles of sodium here. This is a concentration measure. So let me just say for the time being, to simplify it, 142 number of particles present in the extracellular fluid, and only 14 particles present in the intracellular fluid. Again, I'm simplifying it because I have not yet explained what is the concept of osmolarity. I would explain that in a short bit. So more sodium outside, less sodium in. Reason? Sodium potassium pump. So if the sodium potassium pump is doing this function, then what would happen? More potassium inside and less potassium outside. So how much more potassium inside? So if we talk about potassium, we have about 140 milliosmoles potassium here, and we have about how much? About 4 to 5 milliosmoles. So here I would say 4 milliosmoles. So potassium is about 4 milliosmoles here, about 140 potassium, about 140 milliosmoles here. Again, if we said that this is the concentration of particles, then about 140 particles here and about 4 particles here. Again, what is the reason? The sodium potassium pump. So now the question comes, can we explain what's happening here? Can we explain why we have 142 here in the blood vessel and why do we have 140 inside the interstitium? So you might say, well, that simply means that blood vessel is less permeable to sodium, so less sodium moves out. But that's not the situation. Sodium can even move into the cell, so it is really a small ion. So that would not work. So ideally, sodium should be in balance inside the blood vessel and inside the interstitium. In the extracellular fluid, sodium should be balanced. The osmolarity should be balanced. And for all practical purposes, we actually treat it as balanced. I'm just trying to make a very important point. Remember, we talked about in the previous lecture the little piece of ocean which cells have been carrying around them. So here, this is called Donan effect. This is actually the reason. It's not that the cells are trying to have their ocean with them. This is the reason why the difference in compositions is produced. So Donan effect here, what that means is, I'm going to clean up this diagram a little bit. So let's say, let's make this diagram once more. So we have intracellular fluid here, then we have extracellular, I can probably just combine them. And then we have the intravascular compartment, right? So that's what we talked about previously. 3 compartments. So intravascular compartment, interstitial compartment, intracellular fluid compartment. So don't have to write it again. This is extracellular fluid compartment. So now here we were talking about the Donan effect. So here is how it works. Cell doesn't like sodium. Cell actually loves sodium, but it doesn't want to have it inside because it has the sodium potassium pump and it throws the sodium out. So that is an ATP pump. So that explains why sodium is less inside the cell and more outside. Good. Now, in ECF, what is causing the sodium to be in this in this difference? 142 here and 140 here. The reason is the Donan effect. And what is that Donan effect? Donan effect is that proteins which are negatively charged, they tend to attract the positive charges with them. Makes sense, it's natural. Proteins are negatively charged, so they would tend to attract the cations. What are cations? Cations are positively charged molecules or ions. So they would attract the positively charged ions to them. So that means if there were no proteins here, 142 milliosmoles will be here and 142 milliosmoles of sodium will be in the extracellular interstitial fluid. It will be equal. Why the inequality? Sorry. Because the protein is negatively charged, it is pulling the positively charged ions towards it, and due to that there is a relatively greater concentration of the positively charged ions present inside the blood vessels. In the same token, how about the negatively charged things? For example, chloride. Protein is negatively charged, chloride is negatively charged. So protein would try to repel the chloride and chloride would exit out. So chloride would be a little bit sad that, hey, I'm kicked out of the cell, right? Oh, sorry, kicked out of the vascular compartment. So due to this, what would happen is, it's really sad now. So it looks like angry. Well, so the chloride here has been pushed out because of the negative charge of the proteins. So proteins inside the blood vessels are trying to keep the positive ions with them and trying to push the negative ions outside. So that due to that, please remember this, don't forget this, all cations. So cations are positive. How do I remember that? That is funny. I say that cats are positive animals. So cations are the positive ions. All cations are a little bit in larger concentration inside the blood vessels as compared to interstitium. I'm not talking about intracellular fluid, I'm talking about extracellular fluid. So cations are a little bit in larger concentration inside the blood vessels than the interstitium. And the anions, of course the anions, if the cats are positive, then anions are going to be the negative ions. So negatively charged particles are more in the interstitium, relatively more, slightly more than the blood vessels. This effect is called the Donan effect. The same effect, they say, is also established due to the cell. So cell also has the proteins. So remember that resting membrane potential and the equilibrium potentials and those things, that really is something to do with the Donan effect as well. So please don't forget this effect. So anyways, going back to this thing, we were talking about the concentrations. So relatively potassium 4 milliosmoles outside here and 140 milliosmoles over there. If we talk about chloride, about 108 milliosmoles of the chloride outside and about 10 milliosmoles inside the interstitial fluid. So chloride is relatively less here. So chloride is less here. Sodium is less here. Potassium is more here. Potassium is less here. Sodium is more here. and chloride is more here, and so is the situation